Okay, this video is a pre-lab video on the EnzymeLab. Mainly what you're doing here is going to determine what the effect of pH has on sucrase in yeast. Okay, so the objective, students will determine the effect of pH on the yeast enzyme sucrase. Sucrase is what we're going to use to break down sucrose, a sugar. So, kind of getting what we already know, or at least some things you should already know. Okay, there are four biomolecules: carbohydrates, proteins, lipids, and nucleic acids. Starches and sugars are known as carbohydrates. You have monomer or simple sugars that are called monosaccharides, and those monomers are small pieces that make up larger pieces. Those larger pieces called polymers or polysaccharides. Okay, starches would be an example of polysaccharides. Simple sugars like um, glucose, galactose, fructose, those are all considered to be monosaccharides or simple sugars. Those are monomers. So what we're looking here is we can determine the presence of monosaccharides by using a Benedict's test, and mainly it'll be positive for a Benedict's test. Okay. So some new stuff. Sucrose, which is table sugar, is a disaccharide. Di meaning it's composed of two monosaccharides. That's what di means. So in this case, with sucrose, the two monosaccharides that make up sucrose is fructose and glucose. Now, sucrose will result in a negative Benedict's test, while glucose and fructose will both have positive results. Okay, and if we remember, a positive result for a Benedict's test is they turn orange when heat is applied. Okay? So, turns orange when heat's applied. That means it is a positive result. Now, the enzyme sucrase is going to break down sucrose into glucose and fructose. So, pretty much what we're looking at here is the enzyme sucrase is found in yeast. Okay? Well, that yeast enzyme is going to break down sucrose into fructose and galactose. And now, if everything turns out well, if the yeast does its job, then we should have a positive Benedict's test, because the sucrose, which gives off a negative test, will be broken down into two monosaccharides, glucose and fructose, and those two, as seen here, will have positive Benedict's tests. So, your pre-lab. This is your observation, okay, the problem. This is the initial thing, okay. An enzyme found in yeast called sucrase can break down sucrose, a disaccharide, into glucose and fructose, which are monosaccharides. Make sure you write down your observation. Your question: in which pH condition does sucrase work? So here I took the liberty of pretty much giving you your hypothesis. All you have to do is choose the appropriate words. So there's a variety of different hypotheses you can get from this. In fact, you can get 8 different possibilities here. So go ahead and pick your poison here. So you can say sucrose will work in acidic and neutral environments. You can say it will work in basic environments only. You can say it can work in all 3, or it'll work in none. Okay, so whichever your hypothesis happens to be, just go ahead and write it down. To help you out, however, I'll go ahead and tell you that your, not just yeast, but you contain sucrase as well. And sucrase, which is breaking down sugar, okay, your mouth will break down, so that your saliva contains sucrase, as well as some of your intestinal secretions contain sucrase as well. So think about the pH level of those parts of your body when thinking of your hypothesis. Okay, prediction, it's an if-then statement. You know what kind of test you're doing, you know what you're testing, you should be able to determine your prediction. Remember, no personal pronouns, no if my group does this or if I do this. Okay? It's very cold when it comes to doing a prediction or writing anything in a scientific lab report. Okay, no personal pronouns. Just tell me what it is. Don't tell me what you think. Okay, your procedure. So if I were you, I would write these down in your notebook as, word for word. So I'll go ahead and read them all to you. There's 9 steps to this procedure. The first, for every group, you need to obtain 3 test tubes in a test tube rack, some labeling tape, stopwatch, and a graduated cylinder. Okay, that graduated cylinder in your lab will be about a 50 milliliter, but you can use anything as small as a 10. Okay. Number 2, per lab pod. Okay. Obtain a dropper bottle of Benedict solution, a squeeze bottle of activated yeast solution, a squeeze bottle of sucrose A, a squeeze bottle of sucrose N, and a squeeze bottle of sucrose B. A is referring to acidic, um, N is referring to neutral, and B is referring to basic. So number 3, label 3 test tubes, A, N, and B for acidic, neutral, and basic. If I would honestly also make sure you write your initials on there, or 1% of your initials in there, just because you'll probably be putting your test tubes in a beaker of boiling water with other groups. So you may want to put your initials in there so you don't lose track of whose test tubes belong to which group. Number 4, use a graduated cylinder to measure out 5 milliliters of sucrose A solution and add it to test B. Or excuse me, add it to test tube A. All right. Use a graduated cylinder again to measure out 5 milliliters of sucrose in solution and add it to test tube N. Now it's really important that you rinse out your graduated cylinder in between. Okay? If you don't rinse it out, then you're going to contaminate your next solution. So rinse it out after step 4, rinse it out after step 5, and then when you use for step 6, use the graduated cylinder to measure 5 milliliters of sucrose B solution and add it to the test tube. Make sure you rinse it out after every step. It doesn't need to be dried, it just needs to be rinsed out well. Lastly, add 5 milliliters prepared yeast solution to each of the test tubes. Okay, so you'll have a grand total of 15 milliliters of prepared yeast solution, 5 per test tube. Okay, again, rinse out your graduated cylinder when you're done. Wait 10 minutes and then conduct a Benedict's test by adding 15 drops, okay, of Benedict solution to each of the test tubes. Okay, and then I want you to measure in seconds how long it took for the color to change when you added it to the heat. Okay, so you put in your 15 drops, then you add it to a water bath, a boiling water bath. Okay, and there'll be one set out throughout the room. And then as soon as you put it in there, you're going to want, and if I were you, I'd put all 3 of them in there if you can. If you can't, it doesn't really matter. You can do one at a time. But just for time efficiency, you may want to do all 3, or if you can only do 2, then just do 2. You want to clock how long it takes to determine the color change. After about 45 seconds, if the color hasn't changed, it's not going to. So go ahead and pull it out and then record your results. Okay? And then step 9, record your results and then clean up. Your variables and control. So now we're back on the, we're still on the right page here underneath your procedure. So the independent variable, I want you to fill this out. All I'm going to remind you is an independent variable is what you changed during the experiment. Okay, so think about what thing is changed from one tube to the next. Okay, the dependent variable is this is what you're measuring. What are you measuring? Are you measuring time? Are you measuring temperature? Are you measuring pH level? Are you measuring color? What are you measuring? Okay, and then lastly, your control. Now, your control, if I were you, I would just write C, the table. All right, because I do have a control table for you. And tomorrow in class, we're going to fill out that control table. So on the left page of your notebook, at the very top, don't take up too much room, but go ahead and write down this control data table. Okay, 3 columns, 4 rows. So, like I said, it doesn't need to be that big. Just make sure you write it down. And then this is your experiment data table. Okay, so end of 7th period. Um, this is the experiment data table. 4 columns, 4 rows. Make sure you fill them out as is. You're not going to be filling this data table out until you actually do the lab. The control data table that you had on the last slide, we'll be doing that in class tomorrow. Okay? I'm going to check to make sure that both of these tables are written down and your entire right page is done. If your entire right page is not done and you don't have these 2 data tables, you will not complete the lab.